Okay, good morning. We'll get going with this. So a chapter and about a half that I want to get through. We should be able to do this. I'm going to go kind of quickly as we move through here. There are numerous things when I think about Yersinia pestis or the plague. Truly a fascinating pathogen, and the potential for it to cause a handful of different manifestations. We think of bubonic plague with the lymphatics, but septicemic, where we can think circulatory system, and more of a broad involvement with all sorts of organs, and then pneumonic. And the potentials for the plague to be transmitted with all of those mechanisms sets the stage truly for its ability to persist and to really continue to cause issues. Although around here we think pretty low, there are cases of bubonic plague. Lots of times we think kind of the sylvatic cycle, where we think about wild animals, squirrels or other rodents that potentially are harboring it, and fleas as a vector to transmit in all circumstances. But this may be more of an opportunity. The urban rodent flea cycle may be more of the classical bubonic plague situation, where we think about these being a main mechanism, and then others. There are potentials as we think about these arrows for long distance transport that maybe spreads the rodents or ourselves with these pathogens great distances, and that opens up all sorts of other opportunities. So we will see some details with this in a bit. The first bit, thinking bubonic plague, what we're looking at right here, groin, we've got, was that the inguinal lymph node? I think that's what that one is. And it's all swollen up. So this bubo, there are potentials to see, as we see these toes over here that are septicemic plague, these buboes potentially becoming black and necrotic just like this. And plague has been called the Black Death as a result of what we're seeing here. All sorts of, once we think systemic, all sorts of potentials for vascular issues as we see these extremities, the toes, getting impacted, maybe first and then progressing on as we look at that. So here we can see the plague and the Yersinia pestis, the culprit of the plague, in a blood smear, so septicemic situation. These guys are interesting because they have what we call kind of the safety pin appearance. It's bipolar staining. They look gram-positive on the tips. And, you know, if you didn't really know what was going on, you might say maybe that's a spore former, but it's really not a spore-forming bacterium. It's this sort of bipolar staining where the central bit of it typically doesn't pick up that stain. Kind of interesting. So this next one kind of fits, kind of hits close to home, but truly when we think about the incidence of it, it's really not mainly in the Rocky Mountains, but Rocky Mountain spotted fever is the name of this disease, and really identified up in the nose of Montana. Rocky Mountain Labs is a National Institute of Health lab, and there were researchers studying all sorts of tick-borne illnesses and Rocky Mountain, Lyme disease, and a handful of others, relapsing fever, all different tick infections, and a lot of them researched and identified right up in there. So that's really where some of that comes from. So with this, with the spotted fever, the spots are these rashes that appear like these little blotches, petechial rash, and This, unlike the endemic or murine typhus, starts at the hands and then spreads to the trunk, whereas the others, it's the opposite, trunk to the appendages. And we'll talk about these with fleas and lice as culprits for some typhus in a bit. So as we shift over and think Lyme disease, another tick-borne pathogen, there's really some interesting details with this. As we look at this life cycle, it takes 2 years from the time we lay these eggs until we're the adult stage ready to infect maybe us, kind of depends. So we start with this egg. After about a month, we've got this larval stage. It gets onto a host. A mouse is typically what they like. They stay on and attached for about 2 days feeding, taking a good blood meal, and then they overwinter. As we get to the next spring, this larval stage has molted to the nymph stage. The nymph stage gets a host and feeds for a couple of days again, 3 to 4 days, good ample blood meal. Then we molt and become the adult stages. So we see the male and female right up here towards the fall. They get on a host, typically a deer, feed, mate, lay eggs, and there we go. All right, after egg deposition, the tick dies, and then we're back to the start of this cycle. One interesting tidbit that I think is pretty impressive or potentially disturbing, these ticks, the females, are infected and transmit the infection in the eggs. So when the larvae hatch out, they are already infected. If we have this infection going on, every egg is infected, every larva, and in turn, the cycle just keeps going, ensuring that potential. Other animals that become infected with Lyme disease have that potential to serve as a reservoir. And when a naive or uninfected tick happens to take a blood meal, they become infected and the cycle keeps going. So they say Lyme disease results in a characteristic bullseye rash, but that rash isn't something that's seen 100% of the time. It's actually really kind of rare, if you will. Maybe 30 or 40% of the individuals exhibit this. So it's not something that truly everybody that contracts Lyme disease shows. Borrelia burgdorferi is the culprit, the spirochete that causes Lyme disease, and Willy Burgdorfer was the scientist that ultimately identified this as he was doing that research up in Montana. So relapsing fever, Borrelia, a different one, Borrelia recurrensis causes relapsing fever, and it's just a different spirochete, but similar tick transmission as we think about it. So here we can see the deer tick, a big culprit for that, but there are some others that also potentially may be spreading it. But with the tick being a preferred host for that adult stage, it kind of makes sense that the deer tick truly is one of the main culprits with that. So handfuls of different bacteria, anaplasmosis, anaplasma phagocytoileum, flu and fever-like symptoms, and we can see little rodents potentially serving as reservoirs for anaplasmosis. Brucella, brucellosis, Brucella metalensis, abortus, canis, and suis, handfuls of different species. It causes undulating fever. It's a big issue in livestock where we think that abortus causing problems, abortion in livestock, cattle, big concerns. Cat scratch disease, Bartonella hensleyae. This ultimately with cat scratch, cats serving as a reservoir for this, scratches or bites, set the stage to potentially become infected. Lymph nodes swelling, pain, fever, chills. Ehrlichia, this ehrlichiosis, this is from a different tick. This is the Lone Star tick. They've got kind of a little spot or star, if you will, on the back, the abdomen of the tick, hence the name. They also are found in Texas and southern states, but that's not why it's Lone Star. It's because of the little dot on them. And one little fun fact about the Lone Star tick, there is the potential as this tick feeds on you that not only can it give you ehrlichiosis, it also is something that has the potential to make you basically a vegetarian. You become allergic to meat, all protein, like meat protein, that stimulates an allergic response, everything except for primate. So you're not allergic to yourself. And I guess you could potentially, if you really needed a steak, maybe some monkey might be on the menu, I guess. All right. But eating an actual, beef, not something that's going to be good. I have had actually, I think 3 students now that have chimed in with some tidbits. One of them that was last summer that was talking about her mother that lives in Oklahoma or Arkansas. I can't remember exactly. But she developed this from a tick that ultimately she first recognized it eating some chicken, and she got sick, thinking maybe foodborne illness, but truly not. It was allergic reaction and really pretty crazy. Apparently she can eat fish, which made, I'm like, well, isn't that's still meat? I don't know. All right. Odd, but that's what I was told. All right. Okay. And I don't know what's up with this typo. Endocarditis, pericarditis, staphylococcus, streptococcus, enterococcus, and a handful of other hake. This is like just a grouping of some bacilli. We don't need to get bogged down with that. So the septicemia situation where organisms get into the bloodstream through all sorts of opportunities, catheters, dental procedures, piercings or wounds, all have some potentials to bring these into the body and cause endocarditis. The epidemic typhus, here we've got lice that are serving as the vector. Rickettsia prowazaki is the culprit for it. And then the fleas down here for the murine or endemic typhus with Rickettsia typhi. So different rickettsial organisms and different vectors to transmit it. So high fever, aches, rash, potential fatal occurrence with the epidemic. And then the endemic, low-grade fever, rash, headache, and cough, not something that is as serious as the epidemic typhus. The gangrene, we saw some images of this already when we looked at some skin stuff, but it also is important as we get in here with the circulatory, because it is the mechanism that lends to it spreading throughout the body. So definitely issues, rapidly spreading myonecrosis, so necrosis of the muscle cells, edema, yellowish and then purple discharge from the wounds, gas pockets, as we could see with the gas gangrene, and potential for shock and death. Septic arthritis, Staph aureus, and then Neisseria gonorrhoeae are some potentials to cause infectious arthritis or septic arthritis, pain and swelling in the joints. And we see this getting in to the circulatory system through wounds or surgical procedures that may lend to that. I about skipped it. Lyme disease, or Borrelia burgdorferi. There's a handful of different things. So bullseye rash is a potential. Malaise, headache, fever, muscle stiffness. Then early in the dissemination, we see stiff neck, facial paralysis, arthritis, carditis, And then in the late stages, arthritis, meningitis, and possible fatal. There are antimicrobial treatments with that, but oftentimes if they are not given for a lengthy period of time, we set the stage for some of these later stages to progress, lending to bigger problems. And if that bullseye rash isn't apparent in some of those instances, Lyme disease may not be on your radar in the first place. So that lends to maybe not getting the proper treatment that you truly need. I hit typhus already, osteomyelitis, staph, strep, and some others. Here we see bone involvement, osteo, fever, pain, edema, ulcers, and bone loss is a potential. So many. Our plague from Yersinia pestis, like I said, a handful of different potentials with that as far as where we may be seeing issues. It always makes me think of some Monty Python and, bring out your dead. That's what I think of with the plague. Puerperal sepsis, this is something that they've called childbed fever as one potential with that, and Ignaz Semmelweis critical in driving a wedge in the potential for this. Pyogenes and some others are culprits for it, for puerperal sepsis, and I think Streptococcus algalactiae is another one that's a big player. So rapid onset of fever, shock, and death, definitely something serious. And we think about being infected in the course of delivering a child, where we're at greater risk. Rat bite fever, Streptobacillus monoiformis, and Spirillum minor are two of these potentials. So infected rat bites set the stage for this. Rat feces or bodily fluids also can contaminate food or water, lending to transmission that way. Relapsing fever, we showed an image of that with Borrelia recurrensis, Hemsii, and other Borrelia that may set the stage for these recurring fevers and rash that we may see. Streptococcus pyogenes is important when we think about rheumatic fever, among other things, like scarlet fever and others, but circulatory system, this is where we're at with it. So joint pain and swelling, inflammation, heart valves impacted and scarred, and potentially lending to heart murmurs. So this is a consequence of having strep throat and then manifesting elsewhere. Our spotted fever with the rickettsia rickettsii, high fevers and headaches, aches, rash, vomiting, and potentials for fatal hypotension, low blood pressure, as the blood starts to coagulate. That's a serious situation. Toxic shock syndrome is a Staphylococcus aureus, and it's a result of toxins that they can be releasing. If I remember properly, it's a bacteriophage that stimulates the toxin production in this circumstance, lending to the ill effects. There are numerous things that may set the stage for this, a wound lending to the staph, but oftentimes we may think about other things like tampons or other intravaginal products, but tampons were definitely a big one. I want to say it was the 1970s. It could have been a little bit before that. I don't remember all of the details. And being a male, I think I'm fortunate to not have to deal with all of this stuff. But apparently there was a brand of tampon that was being marketed as a new product. You could put one of these in and leave it in for 24 hours or better, super absorbent, and in turn, that was their strategy to market this stuff. And people, I think, thought that's a great idea. And in turn, what really happened was it set the stage for some Staphylococcus to be introduced, and with an ample blood meal in this circumstance and being left in for a good day, That set the stage for the staph to proliferate, to release these toxins, and numerous females started developing toxic shock syndrome, and they're trying to figure out what's going on, and they traced it back to that specific type of tampon. And now the packaging material of that all has some warnings in there talking about the potentials of toxic shock and the need to not leave them in for prolonged periods of time to minimize those potentials. Truly crazy stuff to think about some of that as a potential possibility. Craziness. And also super crazy to think about some of these specifics, like toxins that are only stimulated to be released if that bacterium has been infected with the specific virus, and that virus kicks it into gear and is like, oh, we should start replicating. And in the process of doing that, you're going to pump out some toxins. It's a win for both of us, right? But not a win for us, all right? Toxic syndrome like, or toxic shock like syndrome, Streptococcus pyogenes instead of the staph. And this high fever, vomiting, diarrhea, respiratory distress syndrome, hypoxia, necrotizing fasciitis, and death. Definitely seems a lot worse than the above. They're both truly bad ones, all right? And this is another one that we think of as a potential with streptococcal infection, but in this case, maybe something in the skin or soft tissue infection that lends to it being already, you know, close to the circulatory system that lends to bigger, widespread dissemination. Trench fever, Bartonella quintata, this fever, conjunctivitis, ocular pain, headache, bone ache, shin, neck, and back, all sorts of issues with it. And we think another one for lice serve as a vector. We left off last time with toluramia, our rabbit fever, Francisella tularensis, and rabbits definitely high on the list for potentials to harbor this. So eating or handling an infected rabbit, transmission through tick or fly vectors, also aerosolized transmission. There are opportunities for maybe you're outside doing work and you're mowing a lawn, and there have definitely been instances where a dead rabbit gets ran over with a lawnmower, and that aerosolizes these products and the individual contracts toluramia that way. Crazy, but it happens. There was one where a farmer in Soda Springs, this was maybe 5 years back, was out like mowing brush down. So it wasn't just mowing his lawn, but he was like mowing like, you know, side of the road or whatever. Big, thick growth, didn't see this and hit a rabbit that hit a dead rabbit that ultimately lent to his infection. One that I had a friend that was working in the hospital lab doing micro, and after he had dealt with this, I saw him a month or so later, and he told me, I had a patient with Francisella with tolerantius, and ultimately he had to take antibiotics because they didn't realize that's what they were dealing with. And just as a treatment, everybody, because it is highly contagious, everybody got treated just as a protective. He didn't become infected, but was able to actually identify it, which I thought was pretty interesting. I had another friend that was working in a lab up in Boise and had a patient that he had a blood smear and they had malaria. And he's like, you're in the middle of nowhere, Idaho, right? Thinking malaria, not on the radar. He's like, I see this blood smear and I'm like, I think that's malaria. And he's like, I don't want to be the guy that's like, I got malaria over here. But he calls his supervisor over, and he's like, sure enough, malaria. I'm like, wow, crazy stuff. All right, Burkitt's lymphoma. This Epstein-Barr virus is the culprit for that. That's what we're looking at over here. These abnormal lymphocytes, mononucleosis, is something that we think of as Epstein-Barr that's causing this. And in turn, it can progress to a lymphoma, as we see over here, can. This is cytomegalovirus. Here we can see a kidney cell from a patient with that, and they say it's got an owl eye appearance with this infected cell with the cytomegalovirus. Oh man, as we move in and think about some of these hemorrhagic diseases, Ebola is definitely one that is a terrifying virus. And there are a handful of others that kind of fit right with this, some of these viral hemorrhagic fevers, Marburg and Lassa, along with Ebola, causing some very serious, highly contagious infections. And there's lots of tidbits about some of the Ebola hot zone and some other things spelling out some of the details, and some of it a bit embellished, but a lot of it pretty accurate to think about what's truly happening to these patients, and definitely terrifying. All right, HIV. Here we can see some of the virions budding out of a white blood cell, a T cell. So we're seeing the part of the cycle of this virus. And this showing some global prevalence. I'm not going to get into that stuff right now. There are definitely some countries where prevalence, like East and Southern Africa, where the incidence is really off the charts compared to some other areas where we're fractions of a percentage. Definitely a concern. I'm going to shift from that to this figure because I think it's better to maybe talk about it a little bit like this. This is truly kind of a busy figure as we look at all of these things, but it tells a pretty good story as I will walk you through it. All right. So what we are looking at on here in blue, we are looking at our T cell populations. In red, we're looking at the HIV, the virions or the virus. And we can see both of these interacting over the course of time. I want to point out before I go into it that we start off looking at a handful of weeks, and then we've got a gap in time, and we jump forward and start thinking about years after the initial infection. So 2 different time frames on the same figure. Keep that in mind. On this axis over here is the T lymphocyte population. Over here is the HIV concentration in the plasma. And this is copies of RNA per milliliter of plasma that we're looking at, and number of cells per milliliter of T lymphocytes. All right. So we start off at time 0, the point of becoming infected, and we've got good amounts of T cells in our circulation, little over 1,000 cells per milliliter. All right. So good quantity. As we become infected, we start seeing the level of T cells that are getting infected and getting destroyed as the result of the virus that's targeting them. We see their cell counts start to plummet. After a handful of weeks, we've dropped down to just about 600 or so cells. We've lost quite a few, all right? And the amount of virus has increased pretty dramatically from the point of infection to a handful of weeks into it, all right? As we get about 6 weeks into it, we see a peak with our HIV, and we see a dip with our T cells, all right? This is the acute HIV syndrome. You start feeling a little bit ill. Flu-like symptoms are a lot of the classical initial sort of situation with this. And then it starts to decline, all right? The level of HIV goes down. T cells start to come back up. They never go back to what they were pre-infection, but they come back, all right, a little bit. And we see the level of HIV being kind of held in check the littlest bit. T cells rebound. And then at this point, and it can be a handful of years within here, that we say is the latency period. You kind of feel normal, if you will, all right? Not really recognizing ill effects, all right? It's latent, all right? We see a point, a handful of years into it, where we cross, and the level of HIV surpasses those T cells, and they are plummeting down toward the x-axis at this point, all right? Here's where we start seeing some of the symptoms, all right? It's really at this point, this constitutional symptoms, that we have progressed from talking HIV to now moving into the point of AIDS. So acquired immune deficiency is when our T cell populations have been decimated, and, and we're truly not as effective at stimulating any sorts of immune responses. That's when the opportunistic diseases take hold. We see HIV surging at this point, and they really say, you know, you don't die from HIV, you die from everything else, right? Really, it's HIV that's depleted everything. So I'm like, well, I think that's really the catalyst, right? But truly, it's the fact that we get bombarded with all sorts of other pathogens, many things we've already talked about, and many of which in a normal, competent immune response are something that we would either think of as not really a big deal at all, or something that our body would mount a response against and we'd be good to go. But in these circumstances, you often are infected with handfuls of things that ultimately lend to your demise. There are numerous treatments that we think about now and cocktail approaches to try to drive wedges in the viral replication cycle that in turn dramatically enhances the life of these individuals. And there are definitely some circumstances where we can see levels of HIV being unmeasurable. All right. Does that necessarily mean this patient has been cured? They're technically, they say, unable to transmit. Do they still harbor it? Maybe. All right. I think we're too soon to truly say, yes, they're cured. All right. There have been some other circumstances with some cures, with some with some bone marrow transplants with people that harbor a specific genetic predisposition that renders them immune. We have seen a couple of occurrences with that. As far as vaccine against this, we hope, but currently not something that's available. So as far as the virus goes, there is a whole list of viruses, AIDS and HIV, as we just wrapped up. We start off with flu-like, as I said, and then as we progress to the final stages, all sorts of other circumstances and lots of infections. So we think about contact with bodily fluids, sexual contact, or contaminated needles as a potential to bring it to us. Truly, we need these body fluids. We need access to our circulatory system where those T cells are to lend to the infection. So contact, a hug or shaking hands with someone that has HIV is not good enough to truly infect. We need the interaction of the bodily fluids in order for that to occur. Lymphoma we talked about with our Epstein-Barr. Chikungunya, this virus, fever, rash, and joint pain, it's transmitted by some mosquitoes. The cytomegalovirus we saw in the kidney cell, so asymptomatic, but it can cause non-Epstein-Barr mono, and other issues, developmental issues, or bigger issues in the fetus, and potential for death, transplant rejection, and a handful of other things that may lend to it. Body fluids, blood transfusion, organ transplants, mothers can transmit it to the fetus, either crossing the placenta or through breast milk. It's also transmissible through saliva, numerous ways. And we can see handfuls of different potentials where there's maybe some vaccine or antivirals and handfuls not. Dengue, they call this break bone fever because the pain is so severe that it literally feels as if your bones are breaking. That is terrifying to me. Handful of dengue viruses, different serotypes of those viruses that can result in that, and another mosquito-borne vector. Ebola, all sorts of issues, hemorrhaging basically from any place in the body, and contact with that sets the stage to become contagious. So bodily fluids, sweat, urine, feces, and vomit, all setting the stage for that. Some individuals have contracted and have been cured, and they think, you know, some potentials with serum from these individuals may be harboring some immunity that could be of potential, but still kind of out on that. Hantavirus, we think about it from rodents, mice, and rats, their urine or feces, sending it up aerosolized, inhaling that, lending to infection, and a potential fatal infection with that. Also, some hemorrhagic stuff with hantavirus, dependent upon, you know, some of the manifestations with that, but it's the same rodent feces that lends to that. The mono, like I said, Epstein-Barr or cytomegalo, that may set the stage for that, and also kind of depends on if we're thinking young or adult, but doesn't necessarily always have to be just that way. Yellow fever is where we started this chapter, with that mosquito feeding on the individual. Dizziness, fever, and chills, headache, the potential for some severe liver involvement may be fatal as a potential. Monkeys can serve as reservoirs, and the mosquito serving as some vector. So malaria, Plasmodium, is the parasite for malaria. Mosquitoes serve as the vectors. There are handfuls of malaria species that get us. There are a few others that target other animals. And with that, some of the ones getting us, some of them want to infect young red blood cells, some infect old red blood cells, some of them don't care and they infect any aged red blood cell. That's the bad one. That truly can target any aged cell. That sets the potential for lots of your red cells to be destroyed simultaneously. So the mosquito takes a blood meal, inoculates the parasite into us, it gets into the liver, it replicates from the liver, it's released into the circulation. Then we see the erythrocytic or the red blood cell infection, which is mainly what we think about with malaria. They get into the cells, they replicate, destroying the cells. And it's really as they've destroyed those cells and they're released back out into the circulation where these parasites are able to stimulate an immune response. That's where we see the fevers and chills, the actual symptoms occurring. And then after they've been released from those cells, after a handful of hours, about a day, kind of depends on the specific species, they're back into those red cells, and your symptoms subside as they are hidden away now and continue to infect and destroy. As we've gone through a handful of these rounds of replication, asexual divisions happening in our body, as we've gone through a handful of rounds of these divisions in our red cells, some of those Plasmodium species develop into the sex cells, gametocytes. And these cells are circulating around for a mosquito to tap into us, to take a blood meal, hoping that it sucks up some of these. They reproduce in the gut of the mosquito, make their way through the gut up into the salivary glands. They differentiate into the stage that's infective. We call it a sporozoite, and they're inoculated back into us. That's the life cycle of it in a very quick example. So here we can see some red cells. As they've got in, they've started destroying, we call it a ring form. It forms a big vacuole as it's destroying the hemoglobin. As it starts to replicate, this schizont, this multiple Plasmodium parasites replicating asexually gets to the point that it fills it up and destroys it, as we saw right here, right? The ruptured schizont with all of the merozoites, baby plasmodiums that go on to infect. And the sporozoite, merozoite, these are all different stages of development of that parasite, just different names for them. Toxoplasma gondii, this is another one that has some warning labels. This is on kitty litter, where you may see a warning label that says females especially more of a concern. If you are pregnant or thinking of becoming pregnant, you should use caution when changing the litter box, because the potential to become infected and have it damage the fetus is definitely of concern. In normal situations, people come in contact with toxoplasma. It's almost just a given. And they estimate a good chunk of the population, were we to take a blood sample and test for antibody against it, it's likely we would all test positive, all right? Doesn't mean we're all having serious issues with the parasite. Oftentimes, we don't recognize any ill effects whatsoever. It's really the fetus that's the problem. And if the female becomes infected while cooking a bun up in the oven, that's when the situation truly becomes of concern. The parasite crosses the placenta, infects the fetus, and can cause serious issues, potential stillborn, miscarriage, or severe disease if they actually are born. And that is definitely the big concern. So we think about litter box, but food and water also potential to be contaminated with that, setting the stage for our infection. We do have some other opportunities, but it's mainly the cat that we think of as a big culprit. It can get into animals, forming a cyst, and eating that meat poorly cooked sets the stage for infection. People have also become infected when they have donated blood, and through blood transfusions, individuals have contracted it that way. There are potentials, all right? But truly, the cat is the definitive host. It harbors it, doesn't really recognize ill effects. The feces has the parasite that potentially infects maybe a rodent or a bird, which could also potentially be eaten by the cat, all right? I could see a cat eating a mouse. I could see a cat eating a sparrow. I don't think a cat's going to eat a chicken, but maybe, right? Maybe, all right? Birds, all right? So that sets the stage. And truly, one fun fact with this, our little rodent gets some behavioral shifts as the parasite lodges in the brain and forms a cyst. It makes it really kind of love the cat. It's attracted to the odor of cat urine, and in turn thinks that the cat wants to be its friend. It makes me think of Tom and Jerry. Makes sense. Little Jerry must have had toxoplasmosis. He bugged Tom all of the time, all right? And in turn, the mouse thinks the cat is its friend and tries to get close and befriend it, and the cat eats it. And that's really what happens. Circle of life, right? And in turn, the cat, if it wasn't infected, now it is, all right? There are some thoughts in people where maybe some infection with it, they think might cause maybe thrill-seeking behavior. If you want to bungee jump or something like that, maybe that might be an instance for something like that. There's other potentials for other issues, like mental illness, schizophrenia, and some other potentials that might have toxoplasmosis as the culprit for it. Really kind of crazy. So here's some of the parasites, the tachyozoites, and then the cysts that they can form. A zoetocyst is what they call this, and it's full of these parasites. There is a bit of protective immunity when you become infected with these parasites, and that's why we could see some antibody if we were trying to detect that with people. Babesiosis, Babesia macroti is a parasite that targets us. There are a handful of other Babesia parasites. These are ones that are transmitted through the bite of a tick, and they get into our red cells and infect. Doesn't look the same as Plasmodium, but it's kind of a similar idea with how it's getting into the cells and replicating. Trypanosoma cruzi is a different type of one. Most of the parasites get transmitted through a bite. So the saliva of that insect, as it feeds, it inoculates us. This is the opposite. And these bugs transmit it through their feces. As they are taking a blood meal, they defecate the parasites in the feces. It gets into the wound. It gets brushed into the eye or the mouth. And in turn, the infection results in the human. It causes Chagas disease, and it is something that we think of in southern US and Mexico as we move south. Warmer areas, definitely more of a situation for this. It's transmitted by a triatomine bug. They call it a kissing bug or a cone-nosed bug or cone-nosed beetle because of its mouth parts. This gets me because this isn't 100% true. The assassin bug is not the same as the kissing bug. It's a cousin, all right? They look pretty close, but the kissing bug that transmits Chagas disease, when it takes a blood meal, you don't know it. You don't feel it. When the assassin bug bites you, you know it. It is different, all right? It is a super painful bite, all right? Different type of bug, all right? They look pretty similar, all right? So kind of an issue with that, but I'm gonna let it go. These guys are nocturnal. They're attracted to carbon dioxide, hence the name kissing bug. They crawl up onto your face while you are sleeping. They take a blood meal close to your mouth, and that's how you potentially get infected with it. Kind of crazy. Leishmania, there's a handful of different Leishmania parasites. They can cause skin infections. They also can disseminate to other areas, forming some visceral situations, but those truly are some different species with that, and this cutaneous leishmaniasis. So, handful of different ones that set the stage for that. We didn't talk about any schistosomes. Schistosomiasis is a fluke that gets into us through the life cycle stage of it that's been released out of a snail. It penetrates through our skin and then gets into someplace in the body, hepatic portal veins or the veins feeding into the bladder plexus, laying eggs either into the lumen of the intestine or into the bladder, where we can see them as a potential. Oh man, that's basically it with that. And I'm discard. Pretty much out of time as well. The very last chapter, let me stop this share, start a new share. The nervous system infections, there are a couple of questions on the exam that talk a little bit about nervous system infections, not a huge amount of detail, all right? So a few. Rabies, definitely a big concern. Bacterial meningitis, definitely a big concern, as I skipped over the rabies, all right? Meningitis has numerous culprits, viral, bacterial. There are other potentials. Some fungi might be causing some of these problems. But ultimately, when we think about meningitis, the first thing, we assume it's bacterial. We hope it's not, but we assume it is, because if it is, the potential for fatality is tremendous. So Neisseria meningitidis, the culprit for meningitis. This is the one that we're assuming that it is, but we're hoping it's not. All right. It's a super serious one, lending to definitely some serious mortality rates, if not caught and treated rapidly. All right. So we can see these and some white cells in that circumstance. All right. So there is vaccine against meningococcal meningitis, and definitely something that should be on the radar. We think about close quarters, college dorms, and things like that that may set the stage to greater opportunities of transmission. So whether or not you did get a vaccine against it, something that maybe you should be thinking about. All right. Streptococcus pneumoniae also has potentials for meningitis. We think of it more for pulmonary stuff, but there are some of those opportunities. Got to show some tetanus. This is disturbing. It seems like maybe we need to call in an exorcist, right? Bending away. You really shouldn't be bending. Tetanus causes the rigid body posture, opisthotonus. Basically, your muscles can't quit contracting. We think lockjaw and other situations. If we could see this person's face, he'd be smiling, but he's not happy about it, right? Can't quit grinning, right? Kind of crazy. All right, with that, I will stop. I will tell you, just think a couple of details with some of these tables as you move through here. Like I said, it's just a couple of tidbits of some of these as far as the nervous system goes. Questions or concerns? Good to go? Thank you very much. All right, and my home viewers, farewell to you. It's been a pleasure doing business with all of you. Good day.